Waiting a couple seconds like we've done in the past, just for the sound to kick in. Usually things are pretty good by about now. So today is Thursday, 4/16, and you guys will be watching this video lecture for 7-3 on variations on Mendel. This is going to be a little bit longer one, guys, so I would recommend, I'm just going to make it all one file, one link. But, you know, when you get kind of a good kind of cutoff point halfway through, just stop today and then watch the rest of it tomorrow on Friday. Remember, tomorrow is your quiz 7-2 on Mendelian genetics on section 7-2. I'm just looking at kind of today, and after we have the team meeting, you guys spend a little time watching the video lecture and then save some of it ultimately for tomorrow. So spread it out over a couple of days. Okay, so don't sit down and try to do this all at once. So reminders kind of going back, if you haven't followed Whiteboard at Mr. P or at Whiteboard Mr. P on Twitter, please do so. And then also too, as we move forward with emailing assignments, please put the assignment name and your class period on the email subject line. That just helps me with organization of stuff as it comes in. So it makes things go a little bit faster for me as we move forward. Okay. Pardon me, hopefully the background noise isn't too bad. My kids are playing around kind of upstairs and hopefully they aren't making too much noise. So, so today, looking at section 7-3 on Mendelian genetics. Obviously it's not on tomorrow's quiz, but I wanted to get a jump on it. Tomorrow's quiz on Mendelian genetics. Today we're talking about, variations on Mendel. So let's get right into it today. Okay. So section 7-3, we've kind of made our way through, and we're making our way through kind of currently looking at kind of the basics of Mendelian genetics. But as we move into kind of, this is a little bit beyond basic Mendelian genetics. Genetics is not necessarily just as simple as pure dominance and recessiveness. There are kind of a host of different variations that are found within living things that are actually pretty cool, but it moves a little bit beyond the basics of Mendel's principles. And these are those 6 things listed here. So sex-linked traits, it's called codominance, incomplete dominance, polygenic traits, pleiotropy, and then finally genes and the environment. Okay, so we're going to look through in this lecture these 6 things. Again, I'd recommend maybe kind of get through the first 3 today, the first 3 or 4, and then look at the last 3 or 2 tomorrow. So just kind of divide this up a little bit. Okay. So sex-linked traits. Remember we said, we learned a lot about this last week. Humans, and in our concept questions we worked on last week too. 23 pairs of homologous chromosomes, right? So 46 total chromosomes for that of humans. There are 23 pairs. 22 of those 23 are what are called autosomes. Okay, so these are chromosomes that do not play a role in determining gender. Okay, male versus female. There is 1 pair of chromosomes that humans have. 1 pair is the sex chromosomes, and these are ultimately determined or responsible for determining gender. So females have 2 X chromosomes, okay, and they're called X chromosomes. And then males have an X and a Y chromosome. Okay, so males only have 1 copy of the X chromosome. Excuse me. So the X chromosome has the X chromosome, while these sex chromosomes primarily are responsible for determining gender, they do carry some other genes on them in addition to those that code for gender or sex of humans. So these are what are called sex-linked genes, because these genes or traits are carried exclusively on these chromosomes that are responsible for determining gender. So the consequence of this is that, and we're going to see, is that because males and females have different genotypes, and different females have 2 X chromosomes and males only have one X chromosome. the way in which different some of these different disorders or characteristics that can go along with this are going to be inherited in different ratios because of this difference in the sex chromosomes that are held by males versus females. Okay, so just to give you an image, just again a karyotype of female versus male. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 20. These are all of those autosomal chromosomes. They play no, they are own. They determine other characteristics other than gender. But here are that last pair, that kind of 23rd pair, and that of females, they have 2 X chromosomes, the 23rd pair. Males, they have an X and a Y. So you'll notice that the Y chromosome is quite a bit smaller compared to the X. Okay. So males primarily will have just 1 copy of this X chromosome. Okay. So what's the sort of the consequence of that, right? Is that in cases of sex-linked traits, like color blindness, maybe you know somebody that's colorblind. Maybe you are colorblind. That's not really a big deal. Color blindness isn't like this big, humongous genetic disorder by any means. My father-in-law is colorblind. We see color blindness as something that is more prevalent in males because males only need 1 copy of the color blindness allele to have and be colorblind. Whereas it's very rare for females to be colorblind because females need 2 copies of the allele in order to be colorblind. Okay, so here's an example. So we got a normal vision mother. She's a carrier. She carries 1 copy of the normal allele and 1 copy of the defective allele. And again, colorblind is defective, right? It's not this humongous defect by any means, but and then here's the male over here that has normal vision, right? So there are 4 possible offspring. Okay, so female carrier for colorblindness, and this. Okay, this. Sorry, I'm jumping ahead. My bad. So if we turn this into a Punnett square, here's let's to keep it consistent. Let me just double check. I'm gonna put. Here's the female, the mom along the top. Okay, so she has 2 X's. Okay. And she has the one normal copy. I'll use a capital B. And 1 defective copy. I'll use the lowercase b. On the male side over here, he can pass on in the sex cells he produces an X chromosome that again has the normal allele on it, and a Y chromosome that carries nothing on it for the color blindness gene. Well, the outcomes are, we fill in this capital B, X, capital B, X, capital B, X, lowercase b. So in terms of if they had female offspring, it's impossible for them to have any girls that would be colorblind. They would have offspring that would either be normal vision, have both copies of the normal allele, or they would have females that would still have normal vision, but they would be a carrier. So in this case right here. On the male side of things, though, all it takes is, because males inherit only 1 X chromosome, you would end up with normal vision, normal allele, and then color blindness here, having inherited only 1 copy of the defective allele. So again, you kind of see, you get this feel that color blindness, while is not impossible in females, it is less likely because females have 2 X chromosomes. So therefore, it is masked more easily. Whereas males, we only need to inherit 1 copy of the defective allele, and it's a guarantee that you would have that ultimately result. Okay. So, here's let's say we have a female carrier. She's carrying color blindness and then a colorblind male. So here would be a possibility in which you could end up with a female that is colorblind. So here's the female carrier. She's got normal allele, defective allele using the lowercase. Here's the colorblind male, right? Okay. There is a 50-50 chance if they have a daughter that she could be colorblind. And then same kind of scenario on the male side of things, either a carrier or colorblind for females, or males would either be normal or colorblind. So the reason why I kind of wanted to do this was to say that here's the deal with males. It's impossible to have male offspring, and males can't really be carriers. They either have it or they don't, because they only have one X chromosome. Okay. Another example of like sex-linked traits would be like hemophilia. Hemophilia is again something else that is carried on the X chromosome. Uh-oh. Brecken, are you sneaking down here into class? You want to come say hi?
Hi.
You gotta get in the screen here so they can see.
Hi.
All right. Okay, Dad's got to finish this up, okay?
Okay.
All right, thank you.
Yeah.
Thanks for saying hi. Bye. All right, so hemophilia is another example of a sex-linked trait. It's carried on the X chromosome. What's hemophilia? Hemophilia is it's primarily like a bleeding or blood clotting disorder. People that are hemophiliacs will, they can maybe small cuts or any bruising, or even especially like any sort of internal injuries or internal trauma. They will bleed and maybe even bleed out because they're unable to clot and stop the bleeding with their natural clotting factors in their blood supply. So hemophiliacs have to be real careful about injury or trauma because of their inability to be able to handle or stop their own bleeding naturally, if you will. So same kind of scenario here, right? So here's sorry, it switched a little bit, but here's the female side of it. So, so female side over here. Mom is a carrier for hemophilia, right? Dad on this side does not have hemophilia. So the two outcomes on the female side, right? Female, we could have over here, doesn't have it, normal copies, or is a carrier for it, okay? Again, on the male side here, it's they're gonna not have it or have it. There's no such thing as the carrier concept, right? So same kind of scenario as what we kind of saw with that of color blindness. The fact is that in the same with hemophilia, inheriting these characteristics, it is more likely that you would see it in males than in females. All right, next, codominance. This one's pretty cool. It's where both alleles are expressed, and they're expressed to produce what says right, they're equal phenotypic rates that are found in equal proportions. It's recognized by looking at the heterozygote case. If two distinct phenotypes are seen equally, it is considered codominance. So what do I kind of mean by that, right? Like a case where, oh, dang it. Okay. Sorry, guys. So the heterozygote individual shows or exhibits both of the phenotypes equally. So an example of this would be like blood type. Maybe some of you know what your blood type. There's type A, there's type B, and then there's type AB. Okay, so those that are type AB, those are, let me just reel that back in. Those that are type A blood types, genotype AA, will produce these antigens within their blood supply that only allow them to, say, transfuse if you were to need a blood transfusion, type A. If you're type B, okay, those that have type B blood type, okay, they produce antigens in their bloodstream that only will accept type B. Those that are type AB, okay, they are expressing or creating these antigens within their blood supply in equal amounts of both. Okay, so that's kind of the key case. And when we're talking about the heterozygote individual, it's expression of both. So this is just kind of a little visual, so we understand. So here's type A, produces these surface antigens, and they use the little circle shape here. Here's type B. It has the little sort of blue triangles on the surface of the antigen. The surface antigens represented with these blue triangles. But here's a type AB person, right? Type AB has surface antigens both A and B. So it's expressing in equal amounts both of these particular shapes that help to recognize it as a type AB individual. Okay. And then, well, also this is kind of the cool part. Like, you guys ever heard of, if any of you are type O blood types types will not produce any of these surface antigens. So type O really is considered what's called the universal donor. So the universal donor, if like the American Red Cross ever finds out that you're type O, they will be after you for the rest of your life because they want your blood, because type O can be transfused or used into any individual, okay? Because it does not get rejected. But these other blood types will. Like, you can't put type A blood into a person that's type AB. You can't put type B blood into a person that's type A, and etc. Okay. Another example of codominance to help you guys understand, like in cattle, cattle will sometimes express red hair color versus white hair color. Well, if you take like a red-haired cattle, so RR, and cross that with a white-haired, what you would end up with is a bunch of the heterozygote individuals. So all four of these would be the heterozygote case. What you'd have is not red, not white, but you would have both red and white hair expressed in equal amounts. So again, it's not a mixture. It's not red and white mix. It's just that you would have you'd have equal amounts of red and white hair being expressed within the offspring. So in codominance, right, to sort of sum this up, the offspring, you look at the heterozygote individual for the offspring. In the case of the heterozygote case, the two phenotypic extremes, or the two possible outcomes, are being expressed in equal amounts. It's not a mix or a blending. They're expressed in equal amounts. They're still showing red and white hairs, not a mixture of the two. A mixture is what's called incomplete dominance. So incomplete dominance, the dominant allele does not completely mask the recessive allele. So think of it as, in the heterozygote case, it has one copy of the dominant allele and one copy of the recessive allele. So incomplete dominance is recognized by looking at the heterozygote case again, and you see a blend between the two extremes of the phenotype. So let's look at an example of this, okay? So snapdragons, they're a type of flower, okay? They're like a white, red, or pink flower. And they will show or exhibit incomplete dominance. So red snapdragons, okay? So if I take a red snapdragon that is genotype of having both of the dominant alleles, red dominates over white, and I cross that with a white snapdragon that has both recessive alleles, okay, again, I do this again, what am I going to end up in all four of these boxes? I'm going to end up with capital R, lowercase r, the heterozygote case. What does that look like? It doesn't look like red. It doesn't look like white. If you took red and white paint and mixed it, what would you get? You'd end up with pink paint, okay? So in snapdragons, when you have this case, the heterozygote individual, you end up with a blending between the two extreme ends, okay? So what's going on here is the enzyme responsible for the production of color is produced in smaller amounts in the heterozygous individuals. So it's being that it only has one copy of the dominant allele, it produces proteins that have that reddish pigment, but it doesn't produce as much of them. So which is why, with only one copy of it, which is why it ends up appearing pink. But again, the key here is an incomplete dominance. What do we look for? We look for the heterozygote individual, and it's going to look like a blend, right? A blending between the two extremes. So again, here's sort of a visual to give you that guys an idea. So now let's take two snapdragons that are both pink. So they're the heterozygote individuals. So RR times RR, the heterozygote individuals. So they're both pink snapdragons. So I place those on either side of the Punnett square. Okay. What are the resulting offspring going to look like? 25% of them are going to be red, having inherited both dominant alleles. 25% of them, or 1 out of 4, are going to be white. And then 50% of them are going to be the heterozygote individuals that show that blend or that pink color. Okay. Another example of incomplete dominance, lactose intolerance. Maybe you or some of you know is lactose intolerant. Lactose is a type of sugar that is present within dairy products. And a person with lactose intolerance lacks enzymes to digest different milk products. So they will not create these different enzymes that are good at digesting some of these products that are within milk. So some people can have some milk but do not digest lactose really well. So these are the individuals that are the heterozygote case. Okay, so normal people that are not lactose intolerant, okay, will digest lactose quite normally. Okay, and I imagine that most of you are probably in that case. Okay, there are some people that are completely lactose intolerant, meaning they have both of the recessive alleles, and they really can't eat any sort of dairy products. If you know anybody that's lactose intolerant, ask them about if they've ever eaten some dairy and what happens, right? It's usually not a very pleasant experience, okay, from the gastrointestinal digestive perspective. And then there's some people that are kind of right in the middle. Those that are the heterozygote individuals, people that are considered, a lot of times they're talked about as being lactose sensitive. They can maybe drink some milk products, but they definitely can't eat large amount of milk or milk products. They can maybe have a little bit. It really doesn't affect them because again, they're producing lesser amounts of that enzyme, not as much as a normal person, and certainly not the zero or none at all that a lactose intolerant person produces. Okay, so hopefully that makes sense. Okay. All right, next, moving on, polygenic traits. These are basically this kind of the deal. In Mendel's world that we've been talking about, we've kind of built this impression that you have, one trait, two different alleles, and sort of two different phenotypic outcomes, right? And then we've looked at, incomplete dominance and codominance, and we've kind of seen, equal expression or a blending. Okay, so it kind of builds a little bit on Mendel, okay, but looks at the intermediate, the heterozygote individuals, where it's a little bit unique. The reality of it is there's really, it's a little bit more common that traits are actually carried on more than one gene, and often on more than one chromosome, and that's what polygenic traits are. There is an identification. How do we identify this? It's identified by a continuous variation in a characteristic, and think of sort of a bell curve. So in like the human population, one of the things, and this is why I had you guys do the whole email me your height, because we're going to look at this with your data that you created and the data that I've collected over the years, that in the human population, there is sort of an average, okay, this sort of bell curve. Most of the people in the human population kind of fall in this section, sort of medium or mid-range height. There are very small percentages of individuals that are really, really short, and there are a very small percentage of people that are really, really tall. Think about it, in your average everyday lives, how many times, how often do you see somebody that's, maybe 6'10" or taller? Not very often. Now you go to an NBA basketball game, that's a different story, right? Just vice versa. How often do you see somebody that's maybe, 4'5" or shorter within the human population, or 4 feet or shorter? It's very rare. So these two extremes. And why is that? It's because of the fact that really this characteristic of height actually stems from information carried by more than one gene and on multiple chromosomes. Eye color shows this, things like intelligent weight, skin color, etc. will all express this. Okay, so an example is that of skin color to help you guys understand this. So skin pigment or skin color. It's caused by the amount of melanin pigment produced by cells in the skin. So darker pigments will have a greater amount of, those individuals have a greater amount of these dominant alleles. And then those that are lighter in pigment have lesser amount of these dominant alleles. There's 11 known genes, okay, that are associated with skin color. And each of those 11 genes has a dominant and recessive allele, okay? So those with the least pigment have light skin, and those with more pigment have darker skin, okay? So 11 known genes, each one of those genes having their own dominant and recessive. So, and again, I'm not expecting you guys to sit here and memorize this, but you can kind of get this feel of that there's this, this Punnett square gets quite large, okay? And what do we see? We see that in the human population, this bell curve, right? Most of the human population kind of falls in this sort of middle zone in the bell curve, okay? You know, there are very small percentages of individuals that are really very, very light pigments, and very, very small percentage of individuals that are very, very dark pigmentation. Because again, it's about the fact that the likelihood of receiving none or very small amount of the recessive alleles, or receiving all of the dominant alleles, the probability of that is very slim when you think about it from the two extremes. But the probability of receiving somewhere in the middle here, okay, is greater. And that's kind of in this kind of expanded Punnett square. It's kind of this zone right here in the middle, okay? You kind of get the feel for that. Again, not necessarily asking you guys to recreate this, but I'm using this as just to kind of prove a point and to be able to talk it through. All right, onward and wrapping things up. Another example, pleiotropy. Pleiotropy, it means that one gene has more than one phenotypic effect. So typically, like in Mendel's world, one gene, one phenotypic effect. Okay, if I've inherited the, 2 copies of the dominant allele for purple colored flowers and Mendel's pea plants, then it has one phenotypic effect, purple colored flowers. But in reality, as we find and understand more about human genetics and genetics in general, not just human genetics, the reality of one gene equaling one characteristic is actually very rare. It is more common that one gene actually has an influence upon many characteristics. Not just one thing, but many, many things. A great example of this is sickle cell anemia. So sickle cell anemia is the result of a single abnormal codon within DNA. In other words, there is the 6th amino acid, this little change in the genetic sequence. And the 6th amino acid, it changes from valine to glutamine. And that one little change, it changes the shape of hemoglobin. Now, hemoglobin is so red blood cells are these kind of sort of nice little sort of disc-shaped cells. And hemoglobin is a molecule that is present within these red blood cells. And hemoglobin has a nice shape. And the shape, it's a protein. And again, remember, think Pac-Man's mouth, okay? Well, hemoglobin is really good at recognizing and carrying oxygen, okay? So oxygen will fit into that opening within the hemoglobin molecule that's present within our red blood cells. And now the red blood cell moves its way through its bloodstream and our bloodstream, and then transport oxygen to all parts of our bodies. But with this little change, for those that have sickle cell anemia, again, think about the mouth of Pac-Man. changes. This little sequence of events causes it to be a different shape. So while Pac-Man might be able to be okay with kind of carrying oxygen, it's not going to be as good at it. So this little change causes the red blood cells to actually form a sickle shape. What is a sickle? A sickle is here. We can do this. A sickle is an old farming tool. There it is right there that was used to be able to cut down plants, or to be able to harvest grain and different crops. So a sickle is this sort of funky looking knife thing, right? So the red blood cells will have this sort of sickle shape. So rather than being nice and round like this, they'll kind of be funky and curved. Okay. Well, that's, you might go, well, okay, well, it still can kind of carry some oxygen, blah, 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 but it's got this funky shape. Problem is that if it got this funky shape, it actually it can carry less oxygen. Because it's not a nice round shape and it's kind of like this gnarly shape, they'll sometimes hook onto each other. They'll cause clotting and blood clots. Blood clots can be dangerous. They can cause things like brain aneurysms, strokes. They can accumulate in your spleen. Bottom line is the shape, and the misshapen hemoglobin means less oxygen is transported, means overactive bone marrow, tiredness, dilation of the heart damage. Heart actually enlarges, can lead to heart failure. These blood clots can lead to kidney damage, muscle and joint damage, brain damage, gastrointestinal damage, which is primarily your digestive tract, lung damage. So this one little teeny tiny change, okay, this one little gene, okay, that affects one little outcome becomes not so little, okay? So here's just a kind of a breakdown of what we just talked about. So one little change ends up causing these cells to form this sickle shape, and it leads to all sorts of different phenotypic outcomes, having a wide range, and can be really, really very damaging, can even lead to death for those that have sickle cell anemia, that if they are not monitoring it closely and treating it, okay? So again, pleiotropy is actually quite a bit more common. The old school Mendelian thought of, one gene, one characteristic is, not really true within most living organisms. Whereas I should say, not really true. That's a bad way of me saying it, but it's a little less, it's less common. All right, the last one that I think is really cool is genes in the environment. So there's this idea of nature versus nurture. There's all kinds of interesting genetic tests that are out there. If you guys have heard of the 23andMe packets that you can take, or the genetic testing packets that you can use, and you like a spit test, you send it in, and they'll spit you back different genetic information about you, and they can do different tests for different potentially genetic predispositions that one has, and there's more discussion that we'll have on that later on. But basically, here's the deal. Genes that we've talked about, obviously, they play a role in determining us to a certain degree probability of outcomes. We've worked with these Punnett squares, but it's also found too, okay, that it's not just about genes, but it's also about the environment. These two things play a role in ultimately causing or triggering certain genes to turn off and turn on. And these environmental conditions can create different phenotypes. What do I mean by that, right? Certain cancer genes may run in certain families, or certain genetic disorders that might run in certain families. People might know that a person might be predisposed to have a greater probability of maybe developing a certain cancer or a certain genetic outcome, but it doesn't mean it's necessarily going to absolutely happen. You might have, a brother and a sister, or two brothers or two sisters within the same family that are maybe predisposed to maybe breast cancer. Well, one of them, really, takes care of themselves, really eats right, does all the right things, exercises, and they're going to reduce their potential through their decisions within their environment. They're going to reduce their potential outcome of maybe developing breast cancer. Vice versa, maybe the other sister, she or brother, because again, men can also develop breast cancer, believe it or not. The fact is that they have decided to maybe not take care of themselves as well. Well, that kind of ups the percentage that they may end up developing that form of breast cancer. So environment can trigger different mutations. Environment can turn off or turn on, or I should say, maybe say increase likelihoods or decrease likelihoods of certain outcomes. Because remember, again, genes that we possess it goes back to how we open this unit. It revolves around sort of statements of probability. It is not always an absolute guarantee, right? One cool example of this are in turtle eggs. Turtle eggs actually, when they are laid or born, or however you want to say it, are actually genetically identical. In terms of gender. But when you put turtle eggs within warm sand, or they incubate within warm sand. Hey, Anders, you want to say hi?
Hi.
I'm almost done, buddy. Okay. Right. So warm turtle, if turtles lay their eggs in warm sand, or they incubate within warmer sand, temperature-wise, even though these eggs are genetically identical that have been laid, okay, the resulting offspring will be females. But if those eggs incubate within cooler temperature sands, even though they're genetically identical, they'll develop into males. So this is really a pretty neat thing within nature, almost like a little trick within nature. It goes to show that this idea of environment is much more, and maybe equally as influential as those that of the actual genotype. So the environment greatly changed the phenotype. We get this, even though yes, turtles are different than humans, but we get this from a human perspective. Males and females are different, right? And there are distinct differences between the two. Now, despite the fact that their original genotype was identical, the environment is what caused them to be either male or female. Okay, so just to kind of show you, so this is kind of this idea of, if it's cooler, the percent female is 0%. So cooler, kind of right in here, 0% female, so mainly all males. If it's warmer, okay, 100% female or almost all females. But if it's kind of, if the incubation temperature is kind of somewhere in the middle, sort of in this middle zone, it'll be about 50-50 between the two, okay, as far as the resulting gender of the offspring, okay. Another example of genes in the environment, let's go to this. This is kind of cool. The Mayo Clinic twins study. Not twin. The Minnesota twins study. Minnesota twins study. Nature versus nurture. There it is. Okay. So this is kind of cool. The 1986, the Mayo Clinic underwent and started to track identical twins that had been separated at birth. And the interesting part that was sort of found was that as they tracked these twins that they were separated, or I'm sorry, the University of Minnesota, my bad, it's called the Minnesota Twin Study, that they looked at the course of 20 years. Whoops, what's going on here? And they studied 137 pairs of twins, 81 pairs of identical twins, and that they saw that through time, these twins that were genetically identical, but they had experienced different um, uh, whatchamacallit, they had different environments that they had been raised in. You know, they were separated at birth, raised in different families. They ended up, well, you could, you know, they looked like twins, but there were certainly very distinct differences. They found that maybe one was maybe significantly taller than the other one. One was significantly shorter. One had different, you know, different characteristics. So the finding that nature had a huge role, even though they had the same genetics. Um, and I challenge you guys, even, you know, if you know any identical twins, right? Look closely at them. We have identical twins at Holy Family. And we've had several sets of identical twins at Holy Family over the years. If you look closely, you'll see and you'll notice that there's a little bit of a difference, even though they are genetically identical. You know, they're not experiencing the exact same sort of environment or nature, if you will. I'm sorry, nurture, if you will. They're not experiencing the same environment 24/7 every second of the day. So, you know, the reality of it is that ends up becoming some different phenotypic outcomes, and you can see those slight differences within identical twins. So that was a pretty groundbreaking study, because before that, it had been really thought of, you know, from the Mendel's perspective. You know, one gene, one outcome. The outcome's guaranteed, right? If you have this, it's going to be this. Well, in reality of it, we've found that there's a little bit more to that story. It's about, you know, one gene can have multiple effects, which is more common. And we find that the environment plays a pretty big role on these different outcomes of phenotype, more so than what was initially thought. And we're still, to this day, discovering more and more about what this really means. Okay. All right, I believe that's it. That's where we'll cut it off. So reminding you guys, you know, hopefully, you've kind of looked at this in a couple of parts. You're probably hopefully looking at this on Friday right now. We took the quiz today. Remember, those concept questions are due. And then the genetics packet number 1 is due on Monday. We'll have some time this week in team meetings to look at that, but make sure you're working on that for Monday. Okay. I believe that's it, guys. Remember, you guys know where to find me. If you got any questions, just drop me an email and we'll go from there. Thanks, guys. Bye.